Okay, so I'm going to talk to you today a bit about the ingredients for life on Mars. And specifically, I'm going to be referring to measurements made by the Mars Curiosity rover. There have been many measurements made by other rovers and landers, and these have all sort of put together the full picture of what nutrients, what ingredients are on the surface and in the atmosphere of Mars that could be used by life or could be used as building blocks for prebiotic chemistry. Or, if you want to think of it this way, could be resources for future exploration of life in the form of humans. So our very first touchdown on the surface of Mars was the Vikinglanders in 1976. And I like showing this picture because one of the most interesting things about the surface of Mars was how much it actually looks like parts of the surface of Earth. So on the left you see a photo from Vikinglander, and on the right you see the Atacama Desert in Chile. And so what you see here is very dry, very arid environment. And there's some similarities, and in fact, the Atacama Desert is used as what we call a Mars analog. So it's somewhere we go study aspects of the surface and how things like nitrogen and carbon travel in an environment that has very low amounts of biology, but still manages to have biology that extracts what it needs. So what we know now, in general, sort of the 50,000-foot view of Mars versus Earth, is that on Earth we're warm, we're wet, we have an atmosphere, so we can breathe. And this is because we have a magnetic field. On the other hand, we have Mars, which on the surface is appears to be very much less habitable when we think of life as we know it. And this is primarily because, actually, of the lack of an atmosphere, and that's the lack of the atmosphere stems from the lack of Mars having a magnetic field, or having a negligible magnetic field. So it's thought that Mars lost its atmosphere early on in its history. But through this series of the missions in the last 15 years, we've learned a lot about how to fill in the gaps between early Martian history and what we see on the surface today. Because we think Mars used to be a very different place than it is today. And so as you can see, we've had different strategies over the years. Initially, the first strategy was to follow the water. Life needs water. It needs a solvent. It needs we need water. So the earlier missions in the first part of 2000s were the MER rovers, and these Opportunity is still roving. So this was a great success, and we learned so much about the surface of Mars through these rovers. We've also learned a lot through orbiters and also ground-based observations. Curiosity rover marks the beginning of a different strategy to search for life and the history of possible life. And this is the search, what we call the search for habitability. Oops. So I mentioned that Mars was different in the past. And we've learned this over these series of missions because we've seen rocks, sedimentary rocks that could only be formed in the presence of water. And not only that, but they're formed in water that stands for a long time, okay? And so we have clays. We've also confirmed that there is ice, at least just under the subsurface where Phoenix landed. Well, we know there's ice at the poles, but we also know that in the subsurface of Mars, just under the surface in some places, there is water in the form of ice. We've even seen, with Curiosity, we've seen frost, and Phoenix saw it as well. So there's water on Mars. It's just predominantly in the form of gas and then ice. So it's not in liquid water, and what life really needs is liquid water. So I'm going to specifically focus on the major biogenic elements that constitute the major building blocks of life. There's a lot that can be talked about, and the speakers after me will follow up with a lot of other relevant information on water and on some of the atmospheric chemistry. But I'm going to focus just right now on what we've learned from the Curiosity rover about how carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur are sequestered or held by Martian sediments. So the important thing to think about when we're thinking about these elements is that they're different forms. Like we know, for example, this is atmospheric abundances of these elements. And Sam was following on the back, of course, of Viking, and these atmospheric measurements were actually very similar. So it was great to be able to have two measurements at two different times of the Martian atmosphere. Martian atmosphere is predominantly CO2, so there is plenty of carbon on Mars, but it is locked up in the form of CO2 in the atmosphere. So what we're looking for here is where are the usable elements that could be actually taken up by biology? And so you do have nitrogen. Now, nitrogen on Earth is very important, but it's locked up in the form of atmospheric nitrogen, of N2. Again, you have oxygen. As we know, we really need oxygen to breathe on Earth. At least human beings do. So this is very different than our nitrogen-oxygen atmosphere. But the point of this is to show you that we see these elements on Mars, but what's important is the form these elements come in. So these elements have got to make a habitable environment, to be a true ingredient for life, the ingredients have to come in a form in which they are chemically and biologically available. So what this means is they need to be soluble. They need to dissolve in water and dissociate and be able to do chemistry, or be capable otherwise of movement and be able to be cycled between different forms of elements, either through atmospheric chemistry or hydrology or even biology. And so when we see these elements, when we see different species, when we see, for example, couples of elements such as CO2 and methane, so that's carbon with oxygen and then carbon in what we call a reduced form. When we see nitrogen that's oxidized and nitrogen in the reduced form, and an example is N2O versus NH3. So these indicate that there is a cycle going on, that there is transformation between one form of nitrogen or carbon and the other. And this transformation occurs through geochemistry, through photochemistry, and can possibly occur through biology. So finding the same element in different forms is an indicator that A, it can be used, and B, that it has been used in some way. So they're different pools, and it's been cycled through these pools. So the goal of the Curiosity mission was habitability. And so we take this as, in particular, the instrument that I work on, which is sample analysis at Mars. It's a mass spectrometer. So we're looking at molecular forms, or looking at elements, different elements. So we follow the little acronym for these elements is SHNOPS. So carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And so these are all, we call them biologically important elements because they're incorporated on Earth into things like proteins, nucleic acids, amino acids. So they're all really important, and they can be found in nature as well as in biology. So biology on Earth takes these things from its environment and incorporates it into its metabolism. So this is what we are looking for, in particular on the Curiosity rover. So what is the feedstock? What is actually there? What was there in the past for life to have used? So our target for this mission is Gale Crater. And one of the main reasons we went there is because there's lots of evidence for water. So the orbital, the remote sensing that was done indicated that this was a place where there's layers of sediments, which were laid down by water, and that there was actually inflow of water into this basin, and things like alluvial fans and deltas, which on Earth indicate water running down a slope and then losing its energy and depositing. So we see what we see on Earth associated with water is what we see on Mars. So this is one of the reasons we go there. If you want to look for life, for past life on Mars, you're going to go somewhere where you think water had spent a lot of time. And sure enough, when we got there, we've been able to, in the last 4 years, do a lot of geological exploration. And what we do see is, yes, indeed, that there was a lake, we believe, there's a lake in this basin. So you have lake deposits, which are very fine-grained mudstones, and then you have sandstone deposits. And so what we see is really good proof for water-driven transport of materials, which means materials, including biologically important elements, can be moved around by water. In addition, what we see are these mineral veins. And so mudstones, if you think of like a dried lake. You see mud, it dries, it cracks. And then when it gets wet again, water comes in carrying lots of ions filled with different minerals, and it fills in the cracks. And this is how we get veins, mineral veins. And this is actually what we see on the surface of Mars. We see these mineral veins, which indicate that there is multiple times when water was present in this environment. So you may have had a lake bed that dried out, and then fluids come in with additional nutrients, additional minerals, and deposit. So if we really want to get down deep into what is in the sample, we need to actually take the sample, put it in the body of the rover, and have it go through a series of measurements in our analytical laboratory. Now, the analytical laboratory consists of two main tools. One is a mineralogy tool called Chemin, and it does X-ray diffraction, and it measures the mineralogy. The other tool is sample analysis at Mars. I'll talk a little bit about that, and that measures what molecules are in. So in particular, we're looking for organic molecules, but we're also looking for sulfur molecules, nitrogen molecules. And what we have to do to get there is to drill. We drill, and these are the different sites that we've drilled at. And this is actually our trail, our path. A lot of the work we did initially, we landed at, I don't know if there's a pointer, but we landed at Bradbury Landing. And then we spent a lot of time in that upper right-hand corner at Yellowknife Bay because immediately we saw this evidence of this mudstone, and we got a lot of really good information from our initial samples. So here's the arm of the rover doing some drilling, and we actually drilled the first hole on Mars. So each rover and each mission to Mars is an increase in technology development, an increase in the ability to land something heavier. And in this case, we demonstrated that drilling is possible. Now, we're only drilling a couple centimeters, but this means, we can get drills out there, we can drill through Martian sediments. And so the instrument that we are analyzing our samples with is called Sample Analysis at Mars. And in particular, it's about the size of a large old-school microwave. We're looking for volatiles, so things in gases. When we heat up rocks, what gases come out? We're looking for organic molecules, and we're also looking at isotopic ratios of what comes out, which can tell us about the history of the rocks. The instrument is actually 3 instruments. It's made of a mass spectrometer, a set of gas chromatography columns, which help separate different compounds from one another and help us identify them, and then a tunable laser spectrometer, which is an optical method by which we look at gases and can look at isotopic composition of gases. And this is the tunable laser is what the major instrument we use to look for methane. So briefly, I don't want to get into too much detail, but the process is this. The sample gets scooped, it gets dropped into the left-hand side. That's a sample carousel with about 72 cups. And it gets dropped into this little cup. The carousel turns, and then it pushes the cup up into the oven. Now, there's 2 ovens on SAM for redundancy. It heats the sample, and then the gases are released into the mass spectrometer, but they can also be sent into a trap and then be separated further into the gas chromatograph, or they can be sent to the tunable laser. So it's basically a whole analytical laboratory on Mars, which is pretty exciting. While the size of it may be big for a spaceflight mission, it's actually a whole, if you look on Earth, this suite of instruments would fit inside a room. So some of the results, okay, and you can kind of see some, kind of not see. So what we're looking at here is called evolved gas analysis. On the bottom axis is temperature, and you can also think of it as time, because what we're doing is we're heating the sample up, and it's getting hotter over time, and then it will emit gases. On the left-hand side, you have the signal intensity of the gas. Now, if we just take, for example, water, you can see there's a lot of water that comes off these samples at low temperature. And remember, I was talking about how there is frost. There is water in the form of gas that gets adsorbed onto these samples. And when you heat them, it comes off. And so the main thing I want to show here, this is a sample from Yellowknife Bay, that place we spent a whole lot of time, is evidence for all of these actual elements that are interesting for chemistry and biology. That's carbon, hydrogen, oxygen, nitrogen, and sulfur. So I won't talk as much about oxygen and sulfur, but what we do see is sulfur in an oxidized form and in a reduced form, and it's coming from the minerals. So we have sulfur in abundance just from the mineralogy on Mars. The oxygen there is coming from when we heat up perchlorates, which pretty much blanket the surface of Mars. So this oxygen can be quite destructive. Perchlorates are really reactive, and they produce oxygen, chlorine, all sorts of stuff. So this oxygen in some ways can actually be degrading to organic matter. It can tear it up, and it makes it more difficult to see organic matter in our measurements. However, it does represent the fact that there is oxygen here. And then I'm going to go through each of the other elements individually. So first I'm going to start with hydrogen. And what we see, again, this is another evolved gas analysis on the bottom, and you have water that comes off on low temperature, and then you see on the right-hand side between 600 and 800 degrees this peak in the red curve. And what that's showing, which is really interesting, this is why we have bunches of instruments to basically corroborate each other. So that right upper hand side graphic is showing the X-ray diffraction pattern, and what we see there is a clay, and it's a special clay called a phyllosilicate that holds water or hydrogen in its structure. And in fact, it holds the water that the clay actually formed in. So that's a record from the time the clay formed. So a little bit more about this. The water that you're seeing at the very beginning of the curve is that water in the middle in the dark blue. We call it interlayer or adsorbed water. And that's water that comes off really easily with heating. But then the really interesting water that represents water from the time of formation are those green dots. And that's water that's trapped in the structure in the form of a hydroxyl or an OH molecule. And so that H actually, that was formed, that molecule, when it formed, it kind of encapsulated a lot of information about the environment. And I'm just going to show a quick example of this. What we see here is we were able to, by measuring the water in this clay, we were able to fill in a point in Mars's history. It's really hard to get Martian samples, right? You either have to go to the surface of Mars, you have meteorites, or you're able to make ground-based measurements. And so we have a lot of information, not a lot, we have some information about early Mars, and we have some information from ground-based measurements. But to get the points in the middle is really tough. So what we've been able to do here is by looking at the ratio of heavy hydrogen to light hydrogen, we see information about what the planet was like at the time this clay was formed. And the big picture story here is that the clay formed in water. There was some atmosphere present. So at 3 billion years, Mars had not lost all of its atmosphere or surface water yet. And that's pretty interesting in terms of habitability. Okay, so we move on to nitrogen. We detected nitrate, a nitrogen in the form of nitrate, in surface sediments, and we've seen it in pretty much every sample that we've looked at so far. Now remember, every sample is about 12 samples. So when we pick up or we drill into rock, we see nitrogen. And the important thing about nitrate is that it's a form of fixed nitrogen. So something took, and this happens on Earth through biology and not biology, through abiotic processes. You have a process where nitrogen, the double bond of nitrogen in the atmosphere is broken, and then nitrogen is incorporated in another molecule. And this nitrate is a form of nitrogen that is chemically and biologically available. In fact, it's the form of nitrogen that's in fertilizer. So that's how available it is. And on Earth, nitrogen is so important to metabolism that life evolved the way to break that bond and make nitrogen, or take nitrogen in. So the fact that this is happening, we think, from basically from the atmospheric reactions on Mars, this is very similar to places on Earth that are devoid of life. And these reactions also happen in Earth's stratosphere. So you have these non-biological reactions fixing nitrogen. What this means is it's there on the surface and it's available. Now, I wanted to put this in as well. We don't, the instrument that I work on does not look for phosphorus, but phosphorus is a really important molecule in terms of life, element in terms of life. And so we do have other instruments, ChemIn, the X-ray diffraction instrument, and the APXS, which is the alpha particle X-ray spectrometer. And we can detect, the detections of fluorapatite has been supported. Now, fluorapatite is a form of phosphorus, and it's also the form of phosphorus that's in fertilizer. There is also forms of apatite on Earth that are called biominerals. So phosphorus is really important, especially in the form of phosphate, okay, on Earth for life. So that has been detected also in Martian meteorites. So that's another really important ingredient for life that is on the surface of Mars, locked up in rocks. Okay, so couldn't go through this talk without talking about carbon. And I'm sorry that this is really sort of light, but what we see again is another one of these evolved gas plots, on the bottom with temperature, on the left is intensity. And what you see here is sort of this lumpy plot, right, of carbon. It starts evolving from these rocks in the form of carbon dioxide at maybe 150, 200 degrees, and then it comes off in lumpy peaks and stops coming off around 600, 700. So this is really exciting, right? So we have carbon somewhere in these samples, and we see this with almost every sample we see. But the carbon could have many different sources. It can come from thermal decomposition of carbonates. The tricky thing about that is that, we have this Chemin mineralogy instrument that can detect carbonate up to about 1%. Right around 1% it can't detect. What we see is about 1% CO2. So it's really hard to say. So Chemin doesn't see it, we see it. So we have to think of what else could it be? Well, it could be oxidation of organics. These organics could be Martian in origin. They could be from instrument contamination, or because meteorites have carbon and all bodies in our solar system are bombarded with carbon, with meteorites, you have these extraterrestrial samples bringing in the carbon. So the other really exciting possibility is the decarboxylation or the oxidation of really simple organic acids, such as oxalic acid and acetic acid. And this is really exciting because this is sort of prebiotic chemistry that could be going on the surface, and we could be seeing the evidence of this. It's going to take a little more time and more work in the lab to figure out which of these is the predominant carbon source, but all of these are really interesting. Okay. I'm trying to keep my eye on time here. I wanted to mention the first detection of organic carbon on the Martian surface, and we know this is carbon not from our instrument. This work was published by Carolyn Friesenet in 2016. So chlorobenzene was detected after a lot of work on determining what was background and what was not background. So what we see is at Cumberland, which was a site in the mudstone, we see an increase, a small increase over background over this molecule. And because we have this perchlorate in our sample, what we believe is that this was benzene. You heat this up. I mean, this could be a number of ringed compounds. And when you heat it up, you have it interacting with a perchlorate. And you could have the chlorine substituting for the hydrogen. So you end up with this chlorobenzene. So this is exciting. This is carbon detected on the surface of Mars, not from our instrument background. I briefly want to mention the methane. We did see a methane enrichment above our background. We've seen it once. We are continuing to measure methane. So this methane, it's, and again, like the CO2, it's hard to know the exact source, but we have to think of all of the possibilities. And this graphic, I don't want to go through it too much, gives you most of the possibilities. You can have UV interactions with carbon on the surface to make methane. You can have photochemistry. You can have methane in the form of a clathrate coming up from below. And of course, there's always the possibility of microbial methane coming up from below. So we have to look at all the possibilities, but we don't have enough information to really determine what it is. Okay, so really quick, I'm gonna go through this. So yes, we see sulfur. I'm just gonna check down the list in minerals. We have oxygen, but it's kind of a deterrent to life, in particular to finding life, because when we heat stuff up in the presence of this perchlorate, it breaks down the organic molecules. We have hydrogen in the form of water and possibly organics, and this hydrogen can be implicated as part of the water cycle. There is some cycling of water on Mars, and that will come up in a later talk this morning. Nitrogen, it's there, it's soluble and chemically available. There's not really evidence for anything putting it back into the atmosphere, just the one-way transfer. We see phosphorus in the form of phosphate used for fertilizer, so again, it's available. And then finally, carbon, it's there, and it's probably both organic and inorganic. And we think it also cycles through sublimation. At least CO2 cycles through sublimation. I want to leave a little bit of time for questions, but I did want to just really quick, a footnote on the human habitability of Mars. I found this to be really interesting. I found this on the Journey to Mars NASA site. And so it makes a good point that, as you can see, this is a still from the movie The Martian. And as if you've seen the movie, which you probably have, he grows potatoes in Martian soil with the help of some human byproducts, we'll call it. So, so the point that they're making here is that there are macronutrients and micronutrients and oxygen, carbon, all these things that I just listed, they are in the soil. And yes, that's true, but I would caution us to remember it's about the form that they're in the soil in. It's called, we call the speciation in chemistry. The form of nitrogen, carbon, it all has to be available. And I also want to note that they kind of forgot to say that yes, there is also perchlorate in there. So you'd have to remediate that soil before you used it. And finally, I want to acknowledge everybody on the sample analysis at Mars and MSL teams, NASA Goddard, and everybody here for being an enthusiast and studier or student of Mars. That's it. Is there time for questions? I think there's time for questions. Okay, I see you first.
On a scale of 1 to yes, how badly do you want a Mars sample return mission to do something like that?
Oh yes, we we really want that because we would love to get our hands on Mars and get sediments and stuff into laboratories where you can do a lot more with it than you can over there.
Would you go to one of the past locations you've gone to with Curiosity, or would you try a new location?
That's a really good question, and a lot of people have different thoughts on that. That's hard. I don't know if I can answer that. That's personally, well, because I know the most about Gale Crater, I would maybe go back to that Cumberland site, which is where we see organics, where we see high nitrate. That to me would be very interesting. Okay, see right there?
In light of your research and everything, when the Vikinglanders went and they did their sample and looked for signs of life, it's my understanding that they heated and added water and then looked for signs of life. Would that not have instantly killed anything that had evolved to live in the cold, dry environment there?
So that is possible, but what's also possible, I mean, is whenever you heat something, you make a chemical reaction. You may be killing something, you also may be generating gases. I mean, as you see in this evolved gas analysis, you've got a chemical reactor, basically. And so it's really hard to say whether the reactions you're doing are what you're doing to the sample and how that affects what the sample originally was. I don't know if that answers your question.
Do you feel that the answers they got, I know the answers were kind of ambiguous. They said it could have been an indication of life, or it might have been just an indication of inorganic chemistry or organic chemistry from the samples. What do you think about what they found?
From what I understand now about how perchlorate is so reactive, and particularly in the presence of UV, even without heat, I think it would be very easy for CO2 to be produced by the interaction of perchlorate with whatever is put in there. I'm not going to. I mean, that's only from my experience, from what I know.
One last question. In terms of allocation of scarce resources for future missions, do you think it's likely we will learn more from more? Think it's likely we will learn more from more rovers sampling the soil in different locations, or from a balloon or unmanned aerial vehicle mission that could cover larger distances through the Martian atmosphere?
That's a really good question because it addresses different parts. So what I would like to see, what we don't know about Mars, we don't have spatial resolution. You're absolutely right. A balloon would be able to cover more space. But what we don't know is anything below the first couple centimeters. And because radiation and oxidation is so strong in that first layer, I think the part of Mars that we know least about is what's below the surface. And so the only way that you can get in there is by drilling. And the ExoMars lander has a drill that's supposed to go 2 meters. So I think that's it's important to think about resource allocation, but I think that because we have not been there yet below the surface, that is where I would put my resources.